So if you can find the right microRNA, you have a really powerful way of controlling the activity of many different genes. And this is what we've felt is a really good way to try and treat certain forms of epilepsy.
So who knew that epilepsy is really common in dogs as well as humans? Well, today we have the brilliant physiologist from RCSI in Ireland, David Henshall, who explains to us, in an understandable fashion, his research into microRNA expressions and implementing his findings into a study using them as a treatment for epilepsy in dogs, and even potentially in humans in the future.
Hi Torie, my name is David Henshall. I'm a professor in the physiology department at a university in Dublin called RCSI, University of Health and Medical Sciences. And my job is to study the mechanisms of epilepsy and to try and develop new treatments. That's really the focus of my research. But I also do some teaching at the university. We're a big medical school in the centre of Dublin.
What got you into the epilepsies?
So for my PhD research, I was interested in brain injury and mechanisms of brain injury. And then when I went to do research beyond that, I got an opportunity to work in a different laboratory. And they were curious about whether some of the brain injury mechanisms might be relevant to epilepsy. So they suggested, look, why don't you look at some of these genes, some of these signaling pathways in the brain in epilepsy to see if we can treat epilepsy by blocking some of these injury pathways. So that got me really interested in epilepsy. I began to learn a lot about it, how to study it, how to model it, and that led on then to the research that we're doing now.
So the research that you're doing now, I believe we're involving a species other than Homo sapiens. Tell us about it and what happens,
So what we're about to start doing, or we are now underway doing, is working with pet dogs that have naturally occurring epilepsy. These dogs are actually pets that have very severe forms of epilepsy. A lot of this is very new to me as well, but what I've learned in the last couple of years is that the rates, the frequency of epilepsy in dogs is actually very similar to humans. And in fact, in some breeds, it's much higher than the frequency in humans. So there are some breeds of dogs that have very high rates of epilepsy. And we treat those animals, or vets treat those animals with much the same anti-seizure medicines that we use ourselves to treat epilepsy in humans. Unfortunately, they also have the same problem that many of those anti-seizure medicines do not work in some of these dogs. And if the seizures are frequent enough or severe enough, then the dogs often have to be put down. And this is, of course, absolutely devastating for the pet owners and of course, very sad for the animals.
Tell me about this trial that you are doing with a colleague now who is a vet, Rodrigo. How are things going to work? What's the process?
So we provide them with the experimental medicine, and then they consent the owners of the pet dogs with severe epilepsy to enter into what is a placebo-controlled clinical trial for large animals. So it's run very much like a clinical trial with people, except that it's pet dogs. There are information leaflets that are given to the owners of the dogs, which explain the medicine that we're going to test and why they've been invited to participate in the trial. And that is always because the epilepsy is so severe that really the only other option in the short term is probably euthanasia for the dog. The dog would be normally put down. So we're only talking about animals that are in a very poor state of health already in terms of the severity of the epilepsy, and the involvement in the trial is an option for them if they choose to do so. And of course, nobody's ever pushed into getting involved in the trial. If they agree to the trial, then what happens is the dog will receive the medicine, and then after being monitored very closely by the veterinary team, will go home with the parents of the dog, and then it's the responsibility of the pet owners to then monitor the seizures. So they actually keep a seizure diary in the same way that if you were a patient on a clinical trial for an epilepsy medicine, you'd be asked to keep track of your seizures. So the objective is initially just to see, is it safe in pet dogs, because this is a very different type of medicine to what they will have been on in the past, and secondarily, is it effective? So hopefully we find out initially, is it safe and well tolerated, and if so, does it work? And there may be reasons that it doesn't work. For example, it could be that we didn't quite get the dose right for the dogs, and maybe we have to make adjustments there. But first of all, is it safe?
Could you tell us a little bit about this drug that's going to be used, please?
Yes, so this is quite different to the current anti-seizure medicines that are used to treat epilepsy. Most of those all work in quite a similar way. They basically dampen down brain excitability. And we know that about 1 in 3 people fail to get seizures under control with the current medicines. So many of us are trying to think of different ways to treat epilepsy, and what we've learned over the years is if you look inside the brain of somebody with epilepsy, and we're actually able to do that because, of course, some people with severe epilepsy can go on to have brain surgery, where a piece of their brain may be removed, the part of the brain that's triggering the seizures, and that brain tissue sample can then be studied. And over the years, many teams, mine, but many others around the world, have been able to uncover some of the molecular changes, the chemical changes in brain tissue. And what we've found is that many genes which are normally switched off become switched on, and some which are switched on become switched off. And we're talking about hundreds of gene changes here. So if we're going to try and change the hyperexcitable networks in this brain tissue, we may need to alter the activity of many, many different genes. And the medicines that we have at the moment just don't work that way. So how can we change gene activity? The way that we've been approaching this is for sort of looking for a master controller. And a number of years ago, a molecule called microRNA was discovered, which works just this way. The word RNA you may be familiar with already. So if you've ever had one of the COVID vaccines, one of the main COVID vaccines contains something called mRNA, and that mRNA codes for the spike protein. It teaches your immune system to develop antibodies against it. But there are all sorts of other kinds of RNA that are made within the cell, and they sort of act as messengers between DNA and the machinery inside the cell that makes proteins. So what microRNAs do is they latch onto these signals as they pass from the DNA to the protein making machinery inside cells, and they stick to them a little bit like Velcro. And when they latch on to these messages, it reduces levels of those genes. And the interesting thing with microRNAs is that they don't just stick to one. And most of these microRNAs stick to dozens, even hundreds of the little transcripts that control gene activity. So if you can find the right microRNA, you have a really powerful way of controlling activity of many different genes. And this is what we've felt is a really good way to try and treat certain forms of epilepsy. So we've designed a drug that's a little bit like an artificial form of DNA that basically locks onto this molecule and stops it from working. And so that is the molecule that we're now going to start testing.
How exciting. And just to reassure people who aren't overly familiar with this type of these genetics, can you reassure people that this isn't, going to cause their entire genome to mutate and stuff like that? This is a kind of, I presume, a sort of relatively quiet, targeted type of therapy.
Yeah, so this is not a traditional gene therapy as we think of. So we're not going in and tinkering with your DNA, putting genes in or cutting genes out. What we're doing is working outside of where the DNA is on the messages that pass between DNA to the protein synthesis machinery. And these molecules, this sort of artificial drug that we've developed, they stick around for several weeks, but then eventually cells, break them down and they disappear. So it's non-permanent. And actually, a treatment that's a little bit similar to this is also being being used already in children with certain types of neuromuscular disease. So there's a medicine that's based very much on the same type of chemistry that is being used for neuromuscular diseases in children. So it's, in terms of its safety, this is not the same type of drug as your daily pill, but it's also not permanently changing the genes in the brain. It's sort of a short-term tinkerer.
That sounds rather reassuring. So I suppose if it's a short-term tinkerer, that would also mean that it's not sort of a type of treatment that, one would take once or just for a month or a year, but it's an ongoing sort of treatment, a bit like our current anti-seizure medications.
So this will last a lot longer than our current anti-seizure medicines. So from studies that have been done and some clinical trials, it seems that when this type of treatment, this inhibitor, is given, it can last between 2 to 6 months from a single treatment.
Wow.
So it's not permanent, but it would be a very radical departure from having to take pills 2 to 3 times a day.
Goodness, that does sound rather exciting. Gosh, this could go on to another conversation. So could you just quickly tell us what are the benefits of working with a vet and when it comes to dogs rather than, say, mice or another organism?
The possibility here is that we've got actual natural occurring epilepsy, which is naturally drug resistant in a mammal that has a complex, sophisticated brain. Lower species such as mice and rats are very good models in the early phases of drug development, but then they have limitations. They don't perhaps naturally have epilepsy, so maybe you have to trigger it, and maybe the epilepsy that you're triggering is not quite faithfully matching what is occurring in patients. And their brains are, while reasonable models of the human, they're a big step away, whereas dogs actually have the same sort of convoluted surface of the brain, albeit a smaller brain than humans. So we think that the dog brain is a very good intermediate model on the way to moving towards human trials in the future.
And in theory, one hopes that this will literally benefit the dogs who are receiving this treatment and taking part in the study.
Yeah, and interestingly, so the veterinary neurologist that approach us to do this trial, of course, he's thinking first of all of his patients. And initially, I was first thinking of human patients, but I think we're both sort of crossing over our ideas. It would be phenomenal if from doing this research with the veterinary neurologist, we had a medicine that could be used for dogs. That would be fantastic, as well as being a step towards human trials, which of course is the ultimate objective.
Thank you so much for your time today, David. This is, by the way, everybody, stay tuned because soon, I haven't made him promise, but no doubt David will be joining us to tell us all about this really cool book, which will be coming out relatively soon, and we'll explain a bit more about the history of your work in the epilepsies from whenever to today. Is that right?
That's right.
What a great explanation. But yes. So if anybody would like to get in touch with you and learn more about your work and this study, what should they do, David?
I'm based at something called the Future Neuro Research Centre, which is hosted at RCSI in Dublin. And so we have a website, futureneuroresearchcentre.ie, so you can find out more about the work that we do there and ways to contact me through the research centre. We're also on Twitter and LinkedIn, so check us out that way.
Thank you so much to David for sharing with us his exciting and rather, quite frankly, cool research into epilepsy in both dogs and humans. Do ensure, though, that you tune in next week to hear part 2, where we meet Rodrigo Gutierrez Quintana, the veterinary neurologist from Glasgow University, with whom David is working with on this amazing project.